Hi everyone, this is me Monica Pokrel, a third semester grad student, and today I'll be talking about Pasteurella. Let's begin with the overview of Pasteurella. It is a gram-negative Cucobacillus-shaped bacterium that commonly lives in the respiratory tract of animals. It is an encapsulated pathogen, meaning it has a polysaccharide capsules that helps it avoid being destroyed by the immune system. This capsule is also used to classify the bacterium into five cellular capsular serogroups, that is A, B, D, E, and F, each associated with different animal hosts and disease types. Importantly, Pasteurella is a zoonotic pathogen, which means it can spread from animals to humans. In people, infection often happen after animal bites or scratches, particularly from cats and dogs. It also causes soft tissue infections in human, like cellulitis, and sometimes respiratory infections such as pneumonia or sinusitis, especially in those with weakened immune systems. There are three main subspecies that can cause human disease, such as Pasteurella multocida subspecies multocida, Pasteurella multocida subspecies septica, and Gallicida. Overall, this bacterium's capsule and zoonotic potential make it an important pathogen in both veterinary and human medicine. This slide, we summarize the major toxins and virulence factors of Pasteurella multocida, which contribute to its ability to cause disease in livestock and other animals. First factor is capsular polysaccharide. The bacterium has a polysaccharide capsule classified into serogroups A, B, D, E, and F. This capsule helps it resist phagocytosis and complement mediated killing, allowing it to survive the host's innate immune defenses. Another one is lipopolysaccharide, or also known as LPS. As a gram-negative pathogen, the outer membrane contains LPS, which acts as an endotoxin. It not only triggers inflammation but also helps the bacterium evade immune responses. Third one is adhesins, or outer membrane proteins. Proteins like OMPA, OMPH, OMA87, HSF1, and PTFA help the bacterium attach to host cells, facilitating colonization of the respiratory tract and other tissues. Now the fourth factor is ion acquisition system. Genes such as EXBD, TONB, SDBA, and SDBP allow Pasteurella to scavenge iron from the host hemoglobin and transferrin, which is crucial for bacterial survival inside the host. Another factor is superoxide dismutases, SODs, or protectins. Enzymes like SODA and SODC protect the bacterium from the oxidative burst generated by host immune cells, enhancing its survival during the infections. Another one is toxin, that is PMT, also known as Pasteurella multocida toxin. PMT highlighted is produced by serogroup D strains and encoded by the TOXA gene. It is a key virulence factor in porcine atrophic rhinitis. PMT triggers cytoskeletal rearrangements through RHO-ZPTase, leading to bone and cartilage destruction in the nasal turbinates of the pigs. Other virulence factors are additional genes like PMHAS, NANB, NANH, PFHA, PLPB are associated with colonization and virulence. Studies shows that these genes are more frequent in isolates from diseased animal compared to the healthy carriers. So now these slides explain how Pasteurella multocida is transmitted among animals and occasionally to humans. Mm, Um, the first way is opportunistic pathogens in carriers. It is primarily an opportunistic pathogens found in upper respiratory tract. Many animals such as cattle, pigs, and birds carry it naturally in their nasal passages and tonsil without showing the symptoms. Mm, Another factor is stress and environmental triggers. However, certain stressors like viral infections, overcrowding, transportation, or poor ventilation can weaken the animal's immune system. This allows the bacteria to cause diseases. A classic example is sipping fever or bovine respiratory disease complex in cattle, which often occurs after transport. Uh, the modes of transmission can be such as the bacterium can spread through aerosols of respiratory secretions like nasal discharge or by close contact in confined areas such as feedlots, pig barns, or poultry houses. In bird, it causes avian cholera, and wild birds can spread to its domestic poultry through contaminated water or the environment. Mm, so it also has a potential to cause human infection, but human infection can be occasional, usually after animal bites or scratches from the pets like cats or dogs. It is more common with Pasteurella canis, but Pasteurella multocida can sometimes cause wound infections in people. The bacterium can survive for sometimes in damp environments, in water or contaminated bedding and feed. This means indirect transmission can occur through contaminated materials or surfaces. Uh, in agricultural settings, cross-species transmission is also possible, for example, from birds to mammals, through adaptation to specific host, often determines how severe the infection will be. So at the bottom of the slide, we can see a simple diagram illustrating transmission. A healthy carrier animal under stress like transport become more susceptible, develops a respiratory tract infection, and then can transmit the bacterium to uninfected animals. So this slides outlines how Pasteurella multocida is diagnosed both clinically and in the laboratory. First, clinical signs vary depending on the host species. In cattle, infection often causes pneumonia, fever, nasal discharge, and long consolidation. In pigs, atrophic rhinitis with nasal bowl deformities, sneezing, and nasal discharge is common. In poultry, fowl cholera can result in sudden death, septicemia, and enlarged liver or spleen. Postmortem examinations reveal characteristics lesions. For example, in bovine respiratory disease, we see long consolidation and bronchopneumonia, whereas avian cholera may show hemorrhagic enteritis. Laboratory diagnosis begin with culture. Specimens such as nasal swab, lung tissue, or tonsils are plated on blood or chocolate agar under aerobic or microaerophilic condition. Colonies are small and may have a distinctive mousy odor, but they grow poorly on McConkey agar. Biochemical tests help confirm its identity. uh, Pasteurella multocida are gram-negative, Coccobacilli oxidase positive, catalase positive, and may ferment glucose. Further identifications include serogrouping or capsular typing, usually by PCR or antigenic methods, which can differentiate types A, B, D, E, or F. PCR-based detection of virulence genes such as SGBA, HGB, PTFA, PMHAS, and TOXA can distinguish virulent strains from commensals. Antimicrobial susceptibility testing through disk diffusion or MIC methods is important in agricultural settings where drug resistance may be an issue. Advanced techniques like whole genome sequencing, PFZ, and ICE detection are useful for research or AMR monitoring. Overall, combining clinical assessment with lab-based culture, molecular typing, and susceptibility testing provide a comprehensive approach to diagnosing Pasteurella multocida infections. So this slide highlights the antimicrobial resistance or AMR characteristics of Pasteurella multocida, a significant pathogen in livestock. First, multi-drug resistance or MDR strains of Pasteurella multocida are increasingly reported in livestock settings, especially in cattle feedlots. For example, in North America, isolates resistance to more than 7 antimicrobial rose from 17% in 2011 to 87% in 2016, showing a steep increase over just 5 years. Resistance in this bacteria is often linked to integrative and conjugative elements known as ICS. This ICS carry antimicrobial resistance genes and even heavy metal resistance genes. Importantly, these elements can be transferred horizontally between bacteria, spreading resistance across Pasteurelliaceae species. Common antimicrobials to which resistance has been observed include macrolides, tetracycline, aminoglycosides, sulfonamides, and silindamycin. Specific resistance gene like TATH for tetracycline, ERM, MPH, and MSR for macrolides, and TEM, CTXM for beta-lactams have been documented. The presence of MDR strains indicates heavy selective pressure from antimicrobial use in feedlots whether for therapy, prophylaxis, or growth promotion. This not only complements veterinary treatment but also has a broader implication for human health, as these resistant strains can act as a reservoir, a key consideration under the One Health perspective. So some interesting facts that I find while reviewing this chapter is that the Pasteurella multocida is a common bacterium found in both domestic and wild animals, residing mainly in the upper respiratory tract. In cattle, it inhabits the tonsils and nasopharynx. In pigs, the nasal cavity, and in birds, the upper tract. In agricultural settings, it is a major contributor to disease like bovine respiratory disease in cattle, atrophic rhinitis and pneumonia in pigs, and fowl cholera in poultry. It can survive in damp condition and water for extended period and occasionally infect humans through bites or close contacts. Disease outbreaks are often triggered by stressors like transport, high stocking density, and viral co-infections. Moreover, this Pasteurella multocida may act as a reservoir for antimicrobial resistance genes, linking animal, environmental, and human health, highlighting a One Health perspective. So, this is all about the Pasteurella. Thank you, everyone.